Hello everybody, Dr. Mike here. In this video, we're going to take a look at an introduction and overview of nutrition and metabolism. Now, the first thing I think we should talk about is what we require for survival. Three major things: oxygen, water, and nutrients. Now, without oxygen, we can only survive a few minutes. Without water, we can only survive a few days. And without nutrients, depending on the nutritional status of the individual, we will not survive likely more than 90 days without nutrients. So this then brings us to what we define as a nutrient. So let's write down a definition of what a nutrient is. And what you'll find is that a nutrient is going to be a substance or a chemically defined component of food that we require for survival, growth, and reproduction. This is a nutrient. And there's many subcategories of nutrients. So you've got macro, micro, and essential nutrients, and we'll go through all of those. The first thing is, what do we do generally with these nutrients? Well, we can take a nutrient and we can bring it into a cell, for example, and in the cell we can use it to produce ATP for survival. Or we can take nutrients that are stored in cells and we can use them to build. This is just a very general overview of what we can do with nutrients. Now, if we were to look at the main categories of nutrients, what you're going to find is they will be made up of carbohydrates, including fiber, fats, proteins, minerals, and vitamins. Now, when we take a look at carbohydrates first, carbohydrates are one of our main energy sources. Carbohydrates and fats are our two main energy sources, really important. When I say energy source, I mean we use it to produce ATP. And we know that we can take glucose, which is going to be a breakdown product of carbohydrates, undergo glycolysis, undergo the Krebs cycle, throw some components of the electron transport chain, and produce a whole bunch of ATP, or energy. Now, you've probably heard nutritionally of something called GI, glycemic index. Not gastrointestinal, but glycemic index. Glycemic index. That's GI. You probably see this on food packets, for example. Now, the glycemic index is a measure, it's a system of ranking, basically, of identifying carbohydrates in foods and when they're digested and absorbed, how they can change our blood glucose levels. That's what glycemic index is referring to. So, for example, because it's a ranking or a scale, it's 0 to 100. Okay, so what that means is if you have a GI ranking, for example, of 0 to 55, that's a low GI ranking. If you've got 56 to 69, that's going to be a medium or moderate GI, and anything above 69, so 69 to 100, is going to be a high GI food product. And like I said, what it basically means is this: if you ingest some food, and it's a carbohydrate containing food, if you digest it and absorb it quickly, it's going to quickly increase blood glucose levels. That's going to be high GI. If it's broken down or digested slowly and absorbed more slowly, then it's going to be a low GI. And this is referring to its ability to change the blood glucose levels and therefore alter the effects of insulin release, which is important for diabetics, for example. So that's glycemic index. And so that's one of the main reasons why we use carbohydrates. Fiber within carbohydrates. So carbohydrates that we cannot digest, so indigestible carbohydrates, which include things like cellulose, for example, we don't ingest for its energy. We actually ingest it because it bulks up our stool and it helps promote gut motility, and it can help feed the microflora inside of our gut as well. So for carbohydrates, main energy source, and as fiber to bulk up our stool and help feed that microflora within our gut. What about fats? Well, like I said, fat is another main energy source. But in addition to that, we use fat for insulation. We know that we can store a lot of our fat in adipose tissue, fatty tissue. It surrounds organs, for example, and that allows for structural support and protection and anchorage and insulation to maintain heat. But in addition to that, fat comprises of the membranes of our cells. So really important for cell membranes. Now, if we like, we could include cholesterol here under fats. And cholesterol is also important for cell membrane integrity, but for steroids as well, steroid synthesis. What about proteins? All right, so proteins are important, not so much as an energy source. It can be used as a backup energy source because proteins can be broken down to amino acids and they can be fed into those glycolytic pathways and Krebs cycle as well. But predominantly, we use proteins for cell structure and function. We know that, right? Because, for example, muscle is made up predominantly of proteins. Enzymes provide function within the body. Now, what about minerals? Many different types of minerals, for example. We can have just minerals generally, which can often turn into ions within the body, things such as sodium, magnesium, potassium, chloride, calcium, and so forth. But there's also trace minerals. And these trace minerals are also called trace metals. And this can include things like zinc, for example, iron, fluoride. They are trace minerals because they are required in such small amounts. And then last but not least, vitamins. Now, with vitamins, you can have water-soluble vitamins and fat-soluble vitamins. I'll go through them in a sec, but let's just write them down. Water-soluble, which means it's not stored in the body, and fat-soluble, which means it is stored in the body because the cells of our body are made up of fats, so we can bring them into the cells and store them. Now, I want to talk about what we call essential nutrients because some of these we can produce and make us and synthesize ourselves. Some of these we cannot. They're the essential nutrients. We require them from our diet in order for us to survive. So you can have essential fatty acids, you can have essential amino acids, and vitamins and minerals are also essential, obviously, because we can't synthesize them ourselves. So let's focus on the essential nutrients now. And first focus on our essential amino acids. Now the essential amino acids, again, we can produce amino acids or synthesize amino acids in our body. Some we can't, we must get from our diet. So let's write this up. Essential amino acids. Now there are 9 for an adult or 10 if you are a child or young adult. Let's have a look at these essential amino acids. First one I want to talk about is phenylalanine. Phenylalanine. Now this is important because phenylalanine, well, tyrosine can be produced from phenylalanine, and we need tyrosine for the production of certain neurotransmitters like noradrenaline and dopamine, for example. So phenylalanine, valine, leucine, isoleucine, threonine, methionine, lysine, histidine, and tryptophan. These are the 9, I hope that's 9, 1, 2, 3, 4, 5, 6, 7, 8, 9. These are the 9 essential amino acids. We must get them in our diet. If we don't, we cannot make them ourselves. So they're the essential amino acids. What about the essential fatty acids? Let's write these down here. The essential fatty acids. And again, so you can make other amino acids from these amino acids in the same way that these essential fatty acids, we can make other fatty acids from these. Now the 2 essential fatty acids you need to know are linoleic acid and alpha linolenic acid. And again, other fatty acids can be made from these. All right, now I want to talk about the vitamins. Let's talk about the water-soluble vitamins and the fat-soluble vitamins, what they are quick run through, because again, we can't make them ourselves. We need to ingest them from our diet or from supplements if we can't get it from our diet. All right, let's have a look at our vitamins. When we look at vitamins, let's do water-soluble first. Now again, with water-soluble, you ingest them and you use them. Firstly, what are vitamins? I should probably define that. A vitamin we need in small quantities. We can't synthesize them ourselves, which I've already stated before because it's essential. Small quantities, and usually are coenzymes. So they help support enzymatic processes. They help metabolism, for example. They help really important biological processes. All right, water-soluble. All right, when we look at our water-soluble vitamins, it's obviously going to be vitamin C, really important, but the B vitamins as well. So vitamin B1, which is known as thiamine, B2, which is known as riboflavin, B3, which is niacin, B5, which is pantothenic acid, B6, which is peroxidine, B7, which is biotin, B10, which is, B7's folic acid. Sorry about that. Folic acid. B10, that's biotin. And B12, the cobalamines. All right. They're our water-soluble vitamins. You can ingest them and you'll pee them out if you don't utilize them. You can overdose in them too, so don't forget. Now, if we're looking at our fat-soluble vitamins, for our fat-soluble, we can store these. Easier to overdose too because we can store them, so don't forget. So when we look at our fat-soluble vitamins, we've got vitamin D, E, K, and A. Okay, so vitamin D, that's the calciferols. Vitamin E is the tocophenols. Vitamin K is vitamin K, and vitamin A is the carotenoids or retinoids. And these are our fat-soluble vitamins. Now, when we look at our minerals, the trace minerals, okay, I spoke about other minerals like sodium, potassium, chloride, calcium, and so forth. The trace minerals, that includes iron, that includes zinc, that includes fluoride, for example, but there's 15 of them, and I'm not going to go through all of those. Now, the last thing I want to talk about is calories and kilojoules, for example. So, when we look at being able to utilize energy, because we need to take energy from food, right? Without the energy within food, we're going to not survive. So we need a way of being able to measure energy within food, and this is where calories came from, the term calories. So calories refer to the ability of increasing the temperature. Well, basically, it's how much energy or heat is required to heat 1 gram of water by 1 degree Celsius. It's the energy or heat required to heat 1 gram of water by 1 degree Celsius. You're probably sitting there going, how is that even relevant? What does that even mean? Basically, a guy called Atwood would take food, he would put that food into a container that had water, he would run electricity through that container until the food was fully burned. When that food was fully burned, he measured the temperature change in the water, and that therefore was the calories, how many calories that food contained. It's just a way of measuring how much energy was contained within that food stuff. Now, in Australia, we've moved on from using calories. People still use it, but we've moved on from using calories, and we start referring to kilojoules. So this is because obviously in Australia we use the metric system, right? Meters, kilometers, for example, kilo. And what's the difference here? So 1 calorie, for example, is equivalent to 4.2 kilojoules. All right? And 1 kilojoule is equivalent to 0.2 calories. So this is just, again, it's just another measure of the energy contained in food itself. All right, so I think that's a quick run through or introduction of metabolism and nutrition. Hi everybody, Dr. Mike here. In this video I want to take a look at vitamin D. So remember, vitamin D is one of the 4 fat-soluble vitamins. D, E, K and A. And a vitamin is a nutrient that we require in very small amounts that we cannot produce ourselves, but we need it for normal growth and development. Vitamin D being a fat-soluble vitamin, what that means is it can be stored within our body because the cells of our body are surrounded by fatty layers, and it allows for the movement of this vitamin into those cells easily and therefore easy to be stored. Water-soluble vitamins, however, they often get excreted from the body quite quickly. That's one of the major differences. So when we look at vitamin D, first thing you need to be aware of is that the main way that we get vitamin D is through UV exposure or our exposure to sunlight. So this is going to be the first step in the synthesis of a precursor vitamin D to an active vitamin D. We can get some from the food that we eat from animal and plant products, but it's quite minimal, but I'll get there in a second. So our exposure to UV light is going to trigger the conversion of a cholesterol that's present in our skin, in our epidermis, called 7-dehydrocholesterol. What the UV light does is it turns 7-dehydrocholesterol into something called cholecalciferol. So cholecalciferol will now be floating around in our bloodstream. Now when it's floating around in our bloodstream, it's ultimately going to get to our liver. And in our liver, cholecalciferol, which is also known as vitamin D3, still not active yet. It's inactive. Once it gets to the liver, it comes across an enzyme. Now this enzyme is called 25-hydroxylase. And what it does is it gives a hydrogen to the 25th carbon of this cholecalciferol molecule, this cholesterol molecule. And it turns cholecalciferol into something called 25-hydroxycholecalciferol. 25-hydroxycholecalciferol, which is also known as calcifediol. Now, interestingly, when we ingest vitamin D, so for example, if we ingest it from animal products, what we usually get it from is fish predominantly and liver. This comes in the form of cholecalciferol, or vitamin D3, and therefore it's just going to enter the bloodstream and go via this particular pathway in the liver. If we ingest it through plant products, one of the main ways is UV activated mushrooms. We actually get ergocalciferol, not cholecalciferol. Ergocalciferol, and this is actually vitamin D2. And this will travel to the liver and undergo this hydroxylation through 25-hydroxylase and then go via this calcifediol, which means once this is produced, it then moves to the kidneys. So we've got calcifediol, also known as 25-hydroxycholecalciferol, traveling to the kidneys. It comes across another enzyme in the kidneys, which is called 1-alpha-hydroxylase. And what 1-alpha-hydroxylase does is it gives a hydrogen to the first carbon of this calcifediol. So now we have a molecule which is 1,25-hydroxycholecalciferol, which is a mouthful, but it's also known as calcitriol. Now what we have is an active form of vitamin D. Important point, these enzymes here, Really important because you're always going to go through this process and turn into calcifidiol, this 25-hydroxycholecalciferol. We're always going to go to this point when you're exposed to sunlight, but that means this enzyme is always going to be activated in the liver. But this enzyme in the kidneys, specifically, it's produced by what's called the proximal convoluted tubule cells. This 1-alpha hydroxylase, that needs to be stimulated to be active. Right, so what that means is when you get a blood test for vitamin D, you are not actually testing often, you're not testing calcitriol or 1,25-hydroxycholecalciferol, you're actually calcifidiol, 25-hydroxycholecalciferol, that's the one you're actually getting tested for because you're always going to be producing that at all times. So that's a good indicator of your baseline vitamin D, at least precursor. If you've got a problem with calcium, which I'm going to talk about in a sec, then they may test this one down the track. So this gets activated. Now the question is, what activates this? Well, we'll get there in a second because we need to now talk about what does the active form of vitamin D, calcitriol, do? That will tell us here. What it needs to do is it needs to increase calcium in the blood. It needs to increase phosphate in the blood. Two really important points. How does it do this? Well, it's stimulated if calcium and phosphate levels are too low. And that means the thing that stimulates 1-alpha hydroxylase is going to be low calcium, low phosphate. Now, think about it like this. What happens is low phosphate, that's going to directly stimulate this enzyme to activate. All right. What indirectly stimulates this is calcium, low calcium. So when calcium levels are low, it actually travels to the parathyroid gland. So you've got your trachea and you've got your thyroid that hugs the front of your trachea. Behind your thyroid, you've got the parathyroid gland. So I'm going to draw the back of your trachea. So here's the trachea, there's the rings of your trachea, this is at the back. The parathyroid glands are these glands that are embedded in the larger thyroid gland. And what the parathyroid gland does is it produces parathyroid hormone, PTH. So low calcium triggered parathyroid hormone to be released, and parathyroid hormone is going to trigger 1-alpha hydroxylase. So that means we've now got active vitamin D. Its whole job is to increase calcium because the stimulus was a drop, increase phosphate, its stimulus was a drop. How does it do it? It does it like this. It increases absorption of both calcium and phosphate at the gastrointestinal tract. It increases reabsorption of calcium and phosphate at the kidneys. So throws it back from the tubes of the kidneys back into the blood. Really important, increased absorption, and it also increases osteoclast activity. Osteo means bone, clast means to crush. It breaks bone down to release calcium and phosphate into the bloodstream. Now you may be thinking, wait a minute, if this is breaking bone down and releasing calcium and phosphate into the bloodstream, don't we need vitamin D for strong bones? This seems like the opposite of what we want. All right, important point. If we have vitamin D, both calcium and phosphate will be released into the bloodstream. Calcium and phosphate love to be together. They're partners for life. They love to be together, and when they're together, they precipitate or mineralize into the bone. So you're going to absorb it in the GIT, it's going to be in the blood. You're going to reabsorb it from the kidneys, it's going to be in the blood. Some's going to be taken out of the bone, it's going to be in the blood, it gets together and then goes back into the bone, deposits into the bone, strengthens the bone. That's what we want. Now, important point. If we have no vitamin D and we only have parathyroid hormone, something interesting happens. Calcium increases in the blood, but phosphate gets peed out. That means this connection, this bond of the two is not present, and bones actually get broken down. So without vitamin D and only parathyroid hormone, bones will start to become brittle. All right, Now I want to talk about deficiencies. What happens if we don't have enough? What happens if we have too much? All right, let's have a look. Deficiencies are important. Let me tell you why. Firstly, because 50% of the world's population is deficient in vitamin D, and that's because of their lack of exposure to sunlight. The color of your skin actually changes how much vitamin D you produce. If you have lighter skin, you'll produce 5 times more vitamin D than somebody with darker skin. And this has to do with where the individual is in the world and their exposure to that sunlight. So let's first talk about deficiencies. So a deficiency, what can cause it? Well, lack of sunlight, obviously, lack of UV light. It can be caused if you've got a problem with fat absorption. Now why? What does this mean? Vitamin D is a fat-soluble vitamin. It needs fat in order to be absorbed from the intestines into the lymphatic system, then to go from the lymphatic system into the bloodstream and then to the tissues of the body. If you've got a problem with fat absorption, so what could that be? An issue with your bile, for example, or maybe it's a problem with your intestinal tract. Doesn't allow you to absorb fats. This can have a problem and lead to diminished vitamin D, and maybe you're not getting enough vitamin D from the diet. Now, the main issue here with the deficiency is going to be the exposure to UV light. So the question then is, how much vitamin D should we be having? So somebody under 1 year of age, around about 400 international units. 600 international units if you're between 1 to 70 years, and 800 international units if you're above 70 years of age. So what happens if you don't have enough vitamin D? Well, you can have something called rickets if you're a child, or you can have something called osteomalacia if you're an adult. It's basically the same thing, right? Both are vitamin D deficiencies that result in issues with bone. Now here's the thing, if it happens in a child, their bone has not matured yet. So there's a lot of collagen and not a lot of the hard stuff, the solidified, mineralized bone tissue from the calcium and phosphate. So if somebody doesn't have enough vitamin D and the calcium and phosphate is leaving, it's still collagenous, so it's bendy. So somebody with rickets gets bendy bones because they're a child and the bone hasn't matured. Somebody with osteomalacia, this is an adult. So rickets happens in children, osteomalacia as an adult. The bone has mineralized and has hardened. So what's now happening is you're pulling calcium and phosphate out of already hardened bones. So it doesn't become bendy, it just becomes brittle. And this is what happens in osteomalacia, brittle bones. All right, so the best way to get vitamin D, especially if you have one of these disorders, is through supplementation. Obviously UV exposure, but supplementation as well. Now what this also means is anytime you're exposed or start to ingest supplements, you have the risk of overdosing. Overdosing vitamin D, very bad, very toxic. It's one of the worst types of toxicities you can get from having too much vitamin. So let's have a look at what happens when you have too much vitamin D. So overexposure. Now you're not going to get overexposure vitamin D from sunlight. You will get overexposure vitamin D from supplementation. So what happens is this. You get stones, bones, abdominal moans, and psychic groans. This is an old school way of remembering what happens if you have too much vitamin D. Stones, bones, abdominal moans, and psychic groans. Let's have a look. So, stones, increased amount of calcium, increases the likelihood of calcium-based kidney stones. They're the stones. Bones, we spoke about osteomalacia, right? If you're an adult, which I assume this is going to be. Abdominal moans. Calcium, remember, you're going to have too much calcium floating through the bloodstream. This is the problem with an overabundance of vitamin D. Abdominal moans. Calcium, we need to tell muscle to contract. So it contracts the smooth muscle of the abdomen contracts the smooth muscle, and anytime you have abdominal pain, it's because the muscle is contracting over something. So, contraction and psychic groans. Now what's this referring to? Too much calcium blocks your neuron's ability to fire properly. So you get a depression of the nervous system. Now not depression in the classical sense, but just a depression in the firing of the nervous system. So nervous depression, leading to the psychic groans. So what we've had a look at here is vitamin D, how it's synthesized, what happens if you don't have enough or if you have too much. Hi everybody, Dr. Mike here. In this video we're going to go through 3 of the 4 fat-soluble vitamins, vitamin A, K and E. I've already recorded a video on vitamin D which I suggest you watch. Let's take a look. So let's first start with vitamin A. Vitamin A, which is the retinoids. Now when we look at vitamin A, we can take vitamin A in through plant material or animal material, and predominantly it comes as retinol, retinal, and retinoic acid. Now interestingly, in the body, retinol can turn into retinal, retinal can turn into retinoic acid, and retinal can reversibly turn into retinol. But once it is retinoic acid, it stays as retinoic acid, and we'll talk more about that in a sec. Like I said, we can get vitamin A from our food stuff, so we can get it from plant material, for example. And in plant material, beta carotene is a good example of it. And beta carotene is just 2 retinols glued together. Animal products that we can get it in include things like liver and kidneys, for example, and it can come as retinol, retinal or retinoic acid, but it's esterified with a long chain fatty acid, which is important because we need to play around with its esterification to that long chain fatty acid, either pull it off or put it back on depending on what we want to do with the retinol. So vitamin A, because it's fat soluble, it can be stored, and when we store it, we usually store it esterified with a long chain fatty acid, which is palmitate as the long chain fatty acid. Okay, what does retinol do in the body? So you've probably heard of retinol being really important when we look at the functions, really important for night vision. That's true. Specifically what we're referring to is low or dim light. Its ability to respond to low or dim light and allow for you to be able to see something. It's also really important for maturation and development, so early on. It's important for maintaining healthy skin and mucosa, so mucus lining. And it's important for reproductive health. So for example, for males, it's important for sperm production, and for females, maintaining a healthy placenta. So that means it's important throughout one's pregnancy. All right, let's talk about how all this happens. Let's first start off in a cell of the eye. So let's just say this is a cell of the eye. What is going to happen is we'll take in retinol. It will get turned into retinal. Specifically, it gets turned into 11-cis retinal. And what 11-cis retinal does is it's like Gollum off Lord of the Rings, trying to find its ring. It really wants the ring, and once it gets it, it doesn't want to let go of it, but it's really paranoid of somebody stealing it. So it's very anxious. It's always looking around. Now the ring in this case is going to be something called opsin. So I like to think of the O as the ring. Opsin, binding to this 11-cis retinal, and it actually produces something called rhodopsin. Now rhodopsin is Gollum with the ring. It's having a look, it's waiting around, seeing what's happening. If a little bit of light comes into the eye, it stimulates it, scares it, it throws the ring away, it removes the opsin, it throws it away, And when it gets scared, it stimulates the optic nerve to send a signal to the brain. Now this is happening at low light. We need rhodopsin to see at nighttime in low light. It's all because of vitamin A. If we have no vitamin A in our diet, we have no night vision. Really important. In actual fact, if we have no vitamin A for long periods, we can ultimately go blind. And sadly, 500,000 children around the world, 500,000 children around the world are blind because of vitamin A deficiency. Luckily, we've begun to fortify foods with vitamins like vitamin A, so golden rice, vitamin A fortified rice, to try and prevent, because it's all preventative, prevent this blindness that's occurring. Now, too much or too little can be damaging. Too little can obviously result in reproductive issues. Sterility, for example, and blindness, damage to the skin, because we know that obviously when we look at vitamin A, it's really important for maintaining healthy skin. And so one of the treatment options for acne is retinoic acid, right? All right, importantly, a pregnant woman, they don't want their vitamin A levels too low or too high. Both can be a threat to the pregnancy, which is extremely important. All right, the next thing is talking about what's happening. So this is just happening at the eye. How do we do all these other functions? Well, let's just take another cell of the body, for example. And we know that in the cell of the body, we have a nucleus, and in that nucleus, we have DNA, which needs to be transcribed and then translated into proteins. So again, what happens is retinol enters the cell, turns into retinal, turns into retinoic acid. Retinoic acid enters, I'll just write it as RA, and it promotes transcription of the DNA into an mRNA. That mRNA transcribes itself, translates I should say, into proteins. And it's these proteins, depending on what tissue is doing this role, it's these proteins that may play a role in maturation development. They may play a role in maintaining healthy skin and mucosa. They may play a role in protecting and enforcing the placenta, and also that for sperm maturation. So this is how vitamin A works. Let's now take a look at vitamin K. Vitamin K is really important when it comes to coagulation. So if you cut yourself, the thing that stops you from bleeding all over the place is we have platelets, which plug up the area very quickly, and then we have coagulation factors, which are proteins that are expressed, and they stick to those platelets to form this nice meshwork that stop any further bleeding and also promote regeneration of the damaged tissue. Vitamin K is extremely important in this process. So vitamin K is important for coagulation. Now this is what happens. Again, there is the DNA within our cells, and we can transcribe that into mRNA, and this mRNA is going to be an mRNA for a coagulation factor. Now there's heaps, right? Coagulation factor 4, 5, 9, 10, there's a whole bunch of coagulation factors. That then gets translated into a protein. Here are amino acids. And we know that proteins have side chains, for example. So there's a side chain. So this is a coagulation factor protein that's been expressed. It's been translated. Here it is. There's the side chain. Something needs to happen to it. What needs to happen to this protein is it needs to be carboxylated. Carboxylated. Let's write that down. Simply, we've got to add carbon dioxides to it. Now, if we add carbon dioxides to it, we're adding carbon dioxides to that side chain. And what we end up getting, something that looks like that, and something that looks like that. Firstly, how does this happen? Vitamin K. Vitamin K comes along and it promotes this carboxylation. That's what vitamin K does. It promotes carboxylation, helps add those two carbon dioxides. You're probably thinking, who cares? All right, now that they're added there, and you can see they're negative, they can now bind to platelets to help mesh up, right? Create a meshwork around the damaged area and promote regeneration. This is how it works. You go damaged site, there's a cut. All right, to stop you from bleeding, luckily, our platelets have come along and they've plugged up the area. There's our platelets. When they do this, they become negatively charged. So there's a negative charge associated with them. All right, then we've got this. I'm going to draw this more simply like this. Like that, like that, like that. That is a coagulation factor. We have these coagulation factors here now. But the thing is, they're negatively charged as well. They can't bind, negative and negative can't bind together. We need something that facilitates this binding. Calcium. Calcium comes along. Calcium has two positive charges associated with it. It binds to the platelets, it binds to the coagulation factors and brings them all in. Calcium. So calcium is allowing for this clotting to occur. But vitamin K allowed for the activation of coagulation factors. Do we still make coagulation factors without vitamin K? Yes, you can see that. They just don't become carboxylated and they don't promote clotting. This is important here because if you donate your blood to a blood bank, they give a chelating agent, something that binds to calcium and stops it from doing this to stop your blood clotting in the bag. All right, so that's vitamin K. Last one we need to look at is vitamin E. Now, vitamin E is an antioxidant, also known as tocopherol. Vitamin E, also known as alpha tocopherol. And what you'll find is that it is a potent antioxidant. Now, it's a potent antioxidant for peroxyl free radicals. So now we need to talk about what is an antioxidant, what is a peroxyl free radicals. Okay, antioxidant. It goes against oxidants, things that oxidize. So things that oxidize like to pull electrons off things. Loss of electrons is oxidation. Leo, loss of electrons is oxidation. So an oxidant pulls electrons off things. We don't want to pull electrons off things. We've got cell membranes that are, remember, you've got a cell membranes filled with fatty acids. Fatty acids have negative charges associated with them. So what's going to happen is these free radicals love to come along and steal electrons. It makes the membrane unstable. We don't want this. This is damaging. So we need antioxidants to stop it. Vitamin E does this. Now, peroxidation is when this happens to specifically fatty acids. Perfect. And so what happens is if a peroxyl, which may look like that, comes along, tries to steal an electron, it damages the fatty acid. It then is missing an electron. It wants to go find an electron itself, and it results in this chain reaction of issues of free radicals being produced and so forth. We need to stop it. So vitamin E actually sits in the membranes of cells. And when peroxyl free radicals come along, it comes out and binds to it and stops it from doing this. But now the vitamin E is bound up with a free radical and is unable to do its job. It needs to be freed itself. In order to do this, vitamin C comes along, which will be the focus of another lecture, and frees that vitamin E and neutralizes the free radical. Perfect. That's what vitamin E does. Now, how do we get vitamin E? Vitamin E is really abundant in nuts, for example, and it's highly abundant in seeds, for example, but also liver. Liver is going to be a good source of many vitamins, probably too much vitamins, in all honesty. So this is vitamin E. It's really difficult to OD. It's really difficult to be deficient because it's stored, but it can happen. So here is a quick run through of the fat-soluble vitamins, excluding vitamin D. Hi everybody, Dr. Mike here. It's time to talk metabolism. So you know the three major macronutrients, proteins, fats, carbohydrates. What happens when you ingest them? Let's have a look. So you take in a burger, you take a bite, you chew it up, you break it down, you get those proteins, fats and carbs in your stomach, which then are further digested once they get into your intestines. So now we have proteins, We've got fats being triglycerides, we have carbs being sugars here. Now your intestines are going to further break these components down into their micronutrients. So that means proteins will break down into amino acids, sugars will break down predominantly into glucose, and triglycerides will break down into glycerol and fatty acids. From the intestines, they will then get absorbed into what's called the portal vein. The portal vein is a blood system that takes all these nutrients from the intestines directly to the liver for processing, okay? Now what you can find is that the amino acids, glucose, glycerol and fatty acids all get taken via the portal vein to the liver. So this is a liver cell here, but triglycerides, as glycerol and fatty acids, actually get absorbed into the lymphatic system. Now the lymphatic system will then send glycerol and fatty acids as triglycerides into the systemic circulation. This is the bloodstream that actually goes to the entire body. So that means, unlike proteins and carbs, fats will actually be delivered to the entire body before them going to the liver. Not all of them, but a lot of them. Okay, now let's just say the amino acids have now been absorbed or taken from the portal vein into the liver, glucose into the liver, glycerol and fatty acids into the liver. What happens? Well, if we've just eaten a meal, the idea is that the body wants to store these products. So we can store amino acids as proteins, we can store glucose as something called glycogen. Remember, if you read a biological term and it ends in OGEN, it means stored and inactive. And it can store glycerol and fatty acids as triglycerides or fat. All right, easy. Now, what if we want to use these substances? Well, mainly we want to do it to produce energy. Energy being ATP. How does this work? Well, okay, let's first look at glucose because this is our primary energy source in the body. Glucose will be turned into something called pyruvate. Pyruvate will jump into the mitochondria and turn into something called acetyl-CoA. Acetyl-CoA goes through this cycle called the Krebs cycle or the citric acid cycle. And as it goes through this cycle and turns into a number of products, it releases carbon dioxide as a waste product, but also releases ATP. Bang, we've created some energy. We also create a small amount of ATP when we turn glucose into pyruvate, but not too much. Now, as we turn this acetyl-CoA into energy, it releases some hydrogen. Now hydrogen, remember hydrogen, helium, lithium, beryllium of the periodic table, hydrogen will travel to the membrane of the mitochondria. Remember the mitochondria being the powerhouse. Why? Because of this whole process here. The hydrogen jumps into the membrane where it starts talking with a number of proteins embedded in the membrane. Now with oxygen, so you need hydrogen and oxygen coming together with these transmembrane proteins and it produces ATP. This is called oxidative phosphorylation. This is the electron transport chain. Okay, so with this process here, we create huge amounts of ATP, around about 32 to 36 ATP molecules. Okay, what if we don't have any oxygen, for example? So if we're going for a jog, this process is fine, but let's just say we're doing a 100-meter sprint. Okay, where we need more ATP than oxygen we can get in. That means this process is going crazy, producing all these hydrogen, but we don't have enough oxygen. Oxygen is the rate limiting step here. So this starts to backlog. So what ends up happening is that the pyruvate turns into something called lactic acid. Lactic acid doesn't need oxygen to be produced, and lactic acid can form ATP. That's why after intensive bouts of usually anaerobic exercise, not requiring oxygen, you get that lactic acid. All right. Okay, what else can happen? Well, the triglycerides that are broken up into glycerol and fatty acids, glycerol can jump into this glucose chain here at the level of pyruvate. This is perfect. Fatty acids can jump into this glucose metabolism down at acetyl-CoA. Important point is that once it hits pyruvate, it can turn back into glucose if need be. But once it's gone past pyruvate into acetyl-CoA, it cannot go backwards. Now this is important because if we don't have any glucose in the system, we start to break down triglycerides for energy. The glycerol jumps in here at the level of pyruvate. Wonderful. Fatty acids jump in here, the level of acetyl-CoA. Wonderful. We start producing ATP. But what happens is, because we don't have any glucose, we want to replenish those stores. And oxaloacetate here jumps back and turns into glucose. Pyruvate jumps back and turns into glucose. But all these fatty acids are coming down at the level of acetyl-CoA, so we start to produce too much acetyl-CoA. So the acetyl-CoA starts to back up and produce something called ketones. Ketones are the beta-hydroxybutyrate, right? Acetoacetone, these are the ketones. They can turn into ATP. Perfect. Okay, this is basically the entire system when it comes to metabolism. Now, obviously, I've missed out on a couple of processes, but this is the metabolic processes of proteins, fats, and carbohydrates. Hi everyone, Dr. Mike here. In this video, we're going to take a look at glycogenesis. Now, the word glycogenesis has the prefix glyco, which means glycogen, and the suffix genesis, which means the beginning of. So it's the beginning of glycogen. What is glycogen? It's the stored form of glucose. And what is glucose? Well, it's that monosaccharide, that simple sugar that we use to create ATP. And we know from previous videos that one molecule of glucose can produce around about 34 to 36 ATP molecules. So it's a really efficient way of creating energy. Now, we know that we can take glucose and we can absorb it into the body, and it can go into a number of different cell types to use to make energy. But in what situations do we want to take glucose and store it in the form of glycogen? Well, these are going to be fed states. So just after eating, also known as the absorptive state, 0 to 4 hours, we're going to be taking the glucose we've just ingested and we're going to store it as this glycogen. All right. So we've got glucose. You can see that there's 6 carbon, 12 hydrogen, 6 oxygen, and I'm going to simplify this ring-shaped structure of glucose into this structure. And you can also see that I've labeled the 6 carbons. That's where the first carbon is, second, third, fourth carbon, fifth carbon, and sixth carbon. This is going to be important because when we create glycogen, all we do is snap together glucose. We have to change it a little bit, but basically we're snapping them together to create this molecule that we can compactly store within the liver or the muscle or the kidney. So here's glucose. First step is we need to turn this glucose into something called glucose 6-phosphate. And you're probably aware that glucose 6-phosphate is the first step in the glycolytic pathway. It's also the first step in creating glycogen. How do we do it? We take some ATP, give that glucose one of those phosphates. We now have ADP, diphosphate, adenosine triphosphate to adenosine diphosphate, and we snap that ATP here. Now, which carbon is that? 1, 2, 3, 4, 5, 6. It's on the 6th carbon, so it's called glucose 6-phosphate. Perfect. Now what we need to do is take that phosphate and put it in another position. We do this with another enzyme called phosphoglucomutase. Take it off the 6th position, put it on the 1st position. So there's the 1st position. Now we've got the phosphate there. Now the next step is different. What we get is something called uridine triphosphate. Now, this uridine is telling you that there's a nucleotide. What's a nucleotide? Remember, nucleotides are really important components of DNA. And we've got nucleotides, they can turn into amino acids and proteins. We've got the nucleotide uracil here, and we've got uridine triphosphate, so there's 3 phosphates as well. We take the uridine and 1 phosphate, pop them off, connect it to that phosphate. So now we've got 2 phosphates and the uridine, so it's uridine diphosphate attached to the 1st carbon. There's the uridine diphosphate. And what we're simply left, if we take that off, is 2 phosphates. Okay, so the uridine triphosphate just turns into diphosphates. Okay, and now we've got this uridine diphosphate attached to the 1st carbon of the glucose. We can now snap these together to create glycogen. And what we do is we take an enzyme called glycogen synthase, makes sense, snaps things together. We take a glucose molecule and we pull off the UDP, and it facilitates the linking together at which carbons? At the 4th carbon and the 1st carbon. 4th carbon, 1st carbon, 4th carbon, 1st carbon, and we put this process on repeat. Now you can see that at some points you have different branching, and you can see that the 6th carbon can also bind to the 1st carbon. And then here we've got 4th and 1st. So what type of binding do we have? We've got 1 to 4 binding, and we've got 1 to 6 binding. And what we end up getting over time, over this period, is this very branched molecule that we call glycogen. Now here's an important point. When we store glucose in the form of glycogen, this is how animals store glucose. So animals store glucose in the form of glycogen. Plants store glucose in the form of starch and cellulose. And they are really, really similar to this structure. In actual fact, what you'll find is that starch is nearly identical, it's just not as branched. So it's the same bonds. What you're going to find with cellulose is it's the same 1 to 4 bonds. However, what you're going to find is that nearly every second glucose is flipped upside down. So what that means is this is down here, and this bond like this is up like that for every second one. Now this is important because the enzyme that we use when we are now in a fasting state, a post-absorptive state, and we need to break this glycogen down back into glucose to use for energy, we need to chop these 1 to 4 bonds and 1 to 6 bonds. And we use an enzyme called amylase to do this, right? So when we have glycogen or starch, we can easily chop it up with this amylase. But if we're ingesting cellulose, certain plant material, we don't actually have the enzymes to break these every second bonds where it's flipped upside down. And this is the reason why we can't break down grass, for example, in the form of cellulose, but cows can. They do have the enzyme that can chop this up. So what we now have is an indigestible carbohydrate, because glycogen is a carbohydrate, just a complex sugar molecule, and that ends up becoming fiber. So this is the quick process of glycogenesis. Hi everybody, Dr. Mike here. Let's talk about the amazing process of gluconeogenesis. What's gluconeogenesis? Well, let's break the word up. Gluco means glucose, neo means new, genesis means the beginning of, let's read it backwards, the beginning of new glucose. Basically, this is producing glucose from non-carbohydrate based sources. Now you know that there's 3 macronutrients, proteins, fats and carbs, and we make glucose primarily from carbs. So if we were to make glucose from non-carbohydrate based sources, that's basically making it from the proteins and the fats. So we've got the fats over here as triglycerides, we've got the proteins over here, and we're going to talk about how they can come in to produce glucose. Now, why do we want to produce glucose? A couple of reasons. One, the brain's only energy source is that of glucose. Now you may say to me, what about ketones? I know ketones can fuel the brain. That's true, but it's a backup energy source, and I'll talk about that very shortly. So why do we want to produce glucose? Well, sometimes glucose is low in the body. This can happen in fasting states. So in what we call the post-absorptive state, when we need to increase our blood glucose levels back to that 4 to 6 millimoles per liter blood glucose levels that it should be at. All right. So you already know of glycolysis, where we take glucose, turn it into pyruvate, and that pyruvate then jumps into the mitochondria, turns into acetyl-CoA, and then undergoes the Krebs cycle to produce a whole bunch of ATP, also produces carbon dioxide, also produces hydrogen, which can go to the membrane and undergo electron transport. A lot of this happens under aerobic conditions, so there's oxygen. We also know that under anaerobic conditions, pyruvate can turn into lactate. Right? Now this is reversible, so when the oxygen's back again, lactate turns back to pyruvate. But what you probably didn't know is that lactate seems to be a normal end product of this glycolysis anyway. So we know that lactate is the preferred end product in the liver and in the muscle, even at rest in the muscle, and significantly more so in active muscle. All right? So what we're saying now is, we have a stimulus in our body, and our stimulus is we've got low blood glucose levels. This travels to the pancreas. The pancreas has certain cells called alpha cells. They pick up this change or this drop in blood glucose, and they release a hormone called glucagon. Now the body also releases cortisol and also releases noradrenaline. So glucagon, cortisol, noradrenaline, also known as norepinephrine, are 3 stimulators of gluconeogenesis, this process we're just about to talk about. And again, what's the end goal? Stimulus. Drop in glucose. What's the outcome? An increase in glucose. That's what we want. All right. So let's have a look and see what happens. So the first non-carbohydrate based source that we're going to look at that turns into glucose is going to be lactate. Let's say that our muscles have produced huge amounts of lactate, and now we're bringing it into the hepatocyte, so the liver cell. This lactate jumps in and reversibly turns into pyruvate. Now, we want ultimately to get glucose, but pyruvate can't go backwards. In actual fact, there's 10 steps in this glycolysis pathway to go from glucose to pyruvate, and you can see I've only written a couple. These are the non-reversible, which means all the other steps can go backwards, but here they get caught up. So specific or special things need to happen in order for that to go to that, that to go to that, that to go to that, and we'll talk about it. So pyruvate, lactate turns to pyruvate, pyruvate goes into the mitochondria, turns into acetyl-CoA, usually. But in this case, pyruvate's going to turn into oxaloacetate. Now, oxaloacetate is needed to bind with acetyl-CoA in order for the Krebs cycle to happen, produce carbon dioxide, produce ATP, produce hydrogen. All right? But in this case, oxaloacetate
needs to turn to glucose. And it does it by turning into malate first, and then malate leaves the cell. Now when malate leaves the cell, it turns back into oxaloacetate. So oxaloacetate couldn't leave the mitochondria. That's what I meant, the mitochondria, not the cell. Once it's oxaloacetate, oxaloacetate jumps into this cycle here. And it will go through a couple of backward steps, going back, back, back until it gets caught up at fructose 1,6-bisphosphate. It can't go backwards to glucose 6-phosphate. So we need an enzyme. And this enzyme is called fructose 1,6-bisphosphatase. And what it does is it can turn that fructose 1,6-bisphosphate to one step back. Then it's going to keep going back until, boom, it gets caught up again at glucose 6-phosphate. Glucose 6-phosphate will travel into the smooth endoplasmic reticulum. Here in the smooth endoplasmic reticulum, it's going to come across an enzyme called glucose 6-phosphatase. And what it does is it turns glucose 6-phosphate into glucose. And what this glucose can now do is it can exit the hepatocyte. And what do we get? We get an increase in blood glucose levels. That's exactly what we wanted. So this is just from lactate coming in from the muscle, turns to pyruvate, jumps into the mitochondria and turns into oxaloacetate, which turns into malate, which jumps back out of the mitochondria, turns back into oxaloacetate, jumps into this glycolytic pathway, tries to go backwards but gets caught at fructose 1,6-bisphosphate. Fructose 1,6-bisphosphatase comes along, helps it go backwards, keeps going backwards till it gets caught again at glucose 6-phosphate, which then jumps into the smooth endoplasmic reticulum, hits this enzyme called glucose 6-phosphatase, which then turns it to glucose and it can leave the cell. That's lactate. What happens with fats? Well, we know that triglycerides are made up of glycerol and fatty acids. Now, here we've got a glycerol backbone with 3 fatty acids. That's what a triglyceride is. So you relieve one glycerol and you got 3 fatty acids. Now, glycerol will jump into this process here as well. And again, the same thing happens as what happened with lactate. It goes back all through this process until we create glucose and it's back out of the system. Perfect. So that means the glycerol from triglycerides will turn into glucose if needed under gluconeogenesis. What happens to the fatty acids? Well, the fatty acids, they can jump into the mitochondria and they can turn into acetyl-CoA. Now, acetyl-CoA cannot turn into glucose, but it can turn into something else. We'll talk about that in a second. What about proteins? Well, we've got these amino acids. And amino acids, such as alanine and glutamate, for example, what they can do is they can jump into this pathway at different phases. They can jump in here, and that's going to go through that same pathway before. It can jump here. It can jump down here as well. So we know that proteins and amino acids can ultimately turn into glucose as well. Now, what happens if somebody's a diabetic? Well, if somebody's a diabetic, what can happen is this process of breaking down triglycerides into glycerol and fatty acids occur over and over and over and over and over and over and over again because there's very low levels of glucose and more specifically, very low levels of insulin, almost absent insulin. Absent insulin is a trigger to stimulate this breakdown process. Now, the fatty acids are turning into acetyl-CoA. That's being pumped out. Glycerol is going into this process here, right? So as pyruvate comes down to turn into acetyl-CoA, we've got heaps of acetyl-CoA being made, and in order for that to turn into energy, ATP, it needs to bind to oxaloacetate. But you know that when glucose is low, oxaloacetate leaves the mitochondria to turn into glucose. So there's actually not much oxaloacetate for this acetyl-CoA to bind to. So acetyl-CoA just builds up, builds up, builds up, and they actually stack on top of each other. When they stack on top of each other, they produce something called ketones. 2 major types, beta-hydroxybutyrate and acetoacetate. And what these ketones do is they can leave the hepatocyte, they can go to the brain, and the brain can turn it into glucose for energy. Wonderful. But what can also happen is when you produce ketones, you also produce acid. And an individual with type 1 diabetes who is not being managed with insulin, so the insulin is 0, means they get ketones and acid. That's called ketoacidosis. That's not very good. So what do we have? We've got the process of gluconeogenesis. What triggered it? Low glucose. What was the outcome? High glucose. What else was playing a role? The hormones glucagon, cortisol, and noradrenaline. And what were the substrates for it? Lactate, amino acids, and triglycerides, specifically glycerol.
Hi everybody, Dr. Mike here. In this video I want to take a look at the fed state, also known as the absorptive state, which is what's happening metabolically after we've eaten a meal. So first thing I want to talk about is the fact that the fed state is characterized by high blood glucose levels and subsequently high insulin levels. And the whole point of it is to bring the blood glucose levels back down, and it tells the body to do a couple of really important things. So we've just eaten a meal, and that meal is going to contain proteins, fats, and carbohydrates, which are our macronutrients. They end up getting broken down through our digestive system, predominantly in the mouth, stomach, and first part of the small intestines, known as the duodenum, into glucose primarily, fatty acids, glycerol, and amino acids. Now, the fatty acids and glycerol, which were fats, triglycerides, actually get absorbed into our lymphatic system. So we'll talk about that lastly. Firstly, I want to talk about glucose, and then we'll look at amino acids. So if we have a look at glucose first of all, we've got glucose that's been absorbed from our duodenum, or our small intestines, into our bloodstream. All right. Now, blood glucose levels are going to be high now. Now, if blood glucose levels are high, and it's floating through the bloodstream, it's ultimately going to go to the pancreas and trigger a certain type of cell in the pancreas, known as a beta cell, to release insulin. And what insulin does is something really important. Insulin decreases blood glucose levels by telling certain tissues of the body to open up their transport membranes and let glucose in. That's the first thing. Second thing is insulin inhibits certain functions or metabolic functions of the body, like lipolysis, which is the breakdown of fats to produce energy, and proteolysis or proteolysis, which is the breakdown of proteins ultimately to produce energy. It inhibits this. The reason why is because insulin is here to primarily promote storage. So we've just eaten, we want to store all of these micronutrients and maybe use a little bit for energy once it's inside the cell, because we want to drop this blood glucose level down. So insulin's being released. Now there's certain tissues of the body that are insulin dependent. They need insulin to let glucose in. These tissues include muscle and fat. But tissues like the liver, for example, and the brain and the kidneys and red blood cells and the intestines, they do not require insulin in order to get glucose into their cells. They're insulin independent. And that's important because when our blood glucose levels are high and insulin's released, our brain's always going to take that glucose in. Our liver's always going to take that glucose in, but our muscle and our fat will not. So blood glucose levels have gone up, insulin's been released. This high glucose level, let's look first at what's happening at the liver. Glucose, like I said, will always enter the liver. And once that glucose is in the liver, it can do a couple of things. Primarily, it's going to be stored, and glucose is mainly stored in the form of glycogen. Now remember, if in biology you see a word that ends in O-G-E-N, it means stored and inactive. Glycogen is the stored form of glucose. I like to think about glucose as Lego blocks. Glycogen, when you click all those Lego blocks together, that's how it's stored. This is how most of our glucose is stored in the fed state. Some of this glucose, a little bit less than 10% of it, may turn through a process of glycolysis, going from glucose ultimately into something called pyruvate, and pyruvate ultimately into something called acetyl-CoA. Acetyl-CoA enters something called the Krebs cycle, and in the Krebs cycle it spits out some ATP, so spits out some energy. Really important. Another important point here, however, is the fact that acetyl-CoA can also produce fatty acids. And the process of glycolysis going from glucose to pyruvate, if we need to, it can produce glycerol. And if we bring fatty acids and glycerol together, what do we get? Triacylglycerides. So, triacylglycerides, but we can also get, we can get triglycerides, which is the stored form of fat, and we can also get very low density lipoproteins. Very low density lipoproteins have lots of fatty acids and glycerol, can travel throughout the body and deliver these substances to the adipose tissue, to the muscle tissue, to different tissues of the body. All right, another thing that can happen is, that's glucose, amino acids that we've got can be absorbed in the liver as well, and they can actually just enter the Krebs cycle, this cycle that produces the ATP from acetyl-CoA. All right, what happens to glucose at the muscle tissue? Well, remember, muscle tissue needs insulin. So we've got to write insulin up here because it won't let that glucose in without the insulin. But now the insulin's here, so let's get that glucose in. And similar to what's happening in the liver, glucose can be stored as glycogen. Otherwise, that glucose can undergo glycolysis, turn into pyruvate, go from pyruvate into acetyl-CoA, and that acetyl-CoA can undergo the Krebs cycle again and produce some ATP. The amino acids that are coming in for muscle tissue are predominantly going to be stored as proteins. So you've got protein storage at muscle, glycogen storage at muscle as well. In the liver, you're going to have glucose storage as glycogen, fat storage as well, and amino acids jumping into the acetyl-CoA system. What's happening at adipose tissue? Well, glucose can enter adipose tissue as well. It's insulin dependent, so we need to put insulin here again, like we did for muscle. And then once the glucose enters the adipose tissue, glucose will not be stored as glycogen. What glucose will do is undergo glycolysis, pyruvate, acetyl-CoA, undergo the Krebs cycle, and what's going to happen is that it can spit out some ATP if need be, but will also spit out, let's go this way, glycerol and acetyl-CoA again can turn into fatty acids. We can store those two as triglycerides. But also from the meal that we've eaten. The fatty acids, so we've eaten that meal, the fatty acids and glycerol got absorbed into the lymphatic system, not the bloodstream, right? Not the portal vein, which most of these do, right? Glucose and amino acids, for example. But what happens is fatty acids and glycerol into the portal vein get put together into something called a chylomicron. Fatty acids, glycerol into something called a chylomicron. And that chylomicron can deliver fatty acids and glycerol to the adipose tissue, which again can be stored as triglycerides. Okay? And so what we've got here is a run through of what's happening in the fed state. It's all mediated by this insulin and mediated by high nutrient levels, predominantly high glucose levels, but also high levels of amino acids and high levels of broken down triglycerides in the form of fatty acids and glycerol. Hi everybody, Dr. Mike here. In this video I want to talk to you about the post-absorptive or fasting state. This is characterized by a decrease in blood glucose levels and how the body responds to increase blood glucose levels. Now you may be thinking, why do we focus on glucose? Why aren't we focusing on fats and proteins? And the answer to that question is because glucose is the most important energy substrate for organs such as our brain, for example. Our brain only wants carbs for energy. Now, if you think about other energy sources like proteins and fats and their derivatives like amino acids, fatty acids, glycerol, they all feed into the processes that can turn into glucose or a glucose substrate for energy. So we need to look at glucose itself. Firstly, normal glucose levels, so I'm going to write glucose like this. Normal glucose levels sit between 4 to 6 millimoles per litre. This is where we want it to sit. If it goes too low, blood glucose levels dropping too low, we need to increase it. If it goes too high, we need to bring it back down. What we're going to talk about today is what happens after extended periods of time of not eating, what we call fasting or the post-absorptive state. This can be between meals, so between 4 to 8 hours, or it could even be longer. For example, when we wake up in the morning after not eating for about 10 to 12 hours. What's happening in our body to maintain our blood glucose levels between 4 to 6? All right, so first thing is this. I wake up in the morning, I'm a 70 kilogram male. I have my liver, for example, and in my liver we have cells called hepatocytes, which do all the metabolic processing and a whole bunch of other things. And there's the mitochondria as well, which plays a really important role of producing energy from our micronutrient substrates. We've got our pancreas, which we need to talk about here, and we've also got muscle tissue and adipose tissue as well. All of these organs and structures are playing a role in maintaining blood glucose levels. So I've just woken up. As a 70 kilogram male, I have around about 80 grams of glycogen stored in my liver. Now, firstly, glycogen is the stored form of glucose. So glucose being a simple sugar, if we don't want to use it to make energy or ATP, we click it together like Lego blocks to produce glycogen, and our liver is our primary storage site for glycogen. Our kidney also stores some glycogen that we can use, and I may talk about that in a little bit. So I've got about 80 grams of glycogen stored in my liver, and I start to break it down. Again, I'm not eating. It's been 10 to 12 hours and I haven't eaten a thing. This glycogen, what can happen is it can break down into something called glucose 6-phosphate, and glucose 6-phosphate can reversibly turn into glucose via an enzyme called glucose 6-phosphatase. And that turns into glucose. Glucose can then leave the bloodstream and increase our blood glucose levels. But how does this happen? How do we take stored glycogen, break it down into glucose to be released in the bloodstream? This is where the pancreas comes into play. Because if our blood glucose levels begin to drop, the blood supply that feeds the pancreas stimulates a certain type of pancreatic cell called an alpha cell. And these alpha cells, they produce a hormone called glucagon. Now what glucagon does is it stimulates this process. Glucagon can stimulate glycogen. So if it ends in OGEN, remember it means stored and inactive. That's how you remember glycogen as being the stored and active version of glucose. Glucagon, when the blood glucose levels are low, so that's its stimulus. Its stimulus is when there's a drop in blood glucose levels. Glycogen breaks down glucose 6-phosphate. Glucose 6-phosphatase turns it to glucose. It can then be shuttled out of the hepatocyte into the bloodstream and be delivered to the tissues of the body. But now think about this. Now the glucose released into the bloodstream, what happens to the blood glucose levels? It starts to go up a little bit. We don't want to just continually break down that stored glycogen, right? Because our blood glucose levels will go too high. So the increase in blood glucose will travel again to the pancreas, and this time trigger another cell type called a beta cell, and the beta cells produce something called insulin. And like I said, this is now triggered by an increase in blood glucose. What does insulin do in this process? Insulin again travels to the hepatocyte of the liver, and it's a negative regulator. Glucagon was a positive regulator. Glucagon stimulated this process, insulin inhibited. So now that insulin's been released, it stops this from happening. Blood glucose levels start to drop. If it drops, glucagon stimulates. Goes too high, insulin inhibits. Can you see that this is maintaining a happy, healthy balance? This whole process here that we've just spoken about for the breakdown of glycogen into glucose ultimately, this is called glycogenolysis. Glycogenolysis, which basically means glycogen, there's glycogen, lysis meaning splitting apart or breaking apart. This is the breaking part of glycogen into glucose to make energy. So this is how we first begin to increase our blood glucose levels. However, I only have 80 grams of stored glycogen. Over time, if I don't eat, I will use 8% of my stored glycogen from my liver every hour. Now the kidneys also contribute. The kidneys contribute around about 10% in this process. So don't forget the kidneys, don't discount them. Kidneys contribute 10% of glycogen storage to the utilization to produce glucose in this process. But after 10 to 12 hours of doing this and not eating, my glycogen stores are gone. So how do I maintain blood glucose levels? All right, this is where another process comes into play, which is called gluconeogenesis. Once I've used up my glycogen, the blood glucose levels start to drop again. Glucagon is stimulated. Insulin is inhibited. It's low, glucagon is high, and what glucagon can do is it can travel to distant tissues. So glucagon now can travel via the bloodstream to the muscle. Glucagon can travel via the bloodstream to the fat. And what it does is it's a positive regulator of two important processes. The first process here is proteolysis or proteolysis in muscle. What this does is it takes protein and breaks it down into amino acids. One really important amino acid in this process, which I'll talk about, is alanine. But there are other amino acids, what we call gluconeogenic amino acids, that contribute to this. So glucagon stimulates this. Now think about this. If insulin is released into the bloodstream, doesn't matter how much, even if it's a little bit, this is significantly inhibited. So we need to have low blood glucose levels and low blood insulin levels in order for glucagon to stimulate muscle to break down protein into amino acids and to stimulate fat or lipids or triglycerides to be broken down into fatty acids and glycerol. Because as we know, triglyceride, 3 fatty acids, 1 glycerol. So now what we've done is we've used up our glycogen. Insulin levels are low. Glucagon levels are high. Proteolysis has been stimulated. Amino acids are released like alanine. What we've got here, I didn't talk about it, is called lipolysis, I should probably say. Lipolysis, which is the splitting apart of triglycerides into fatty acids and glycerol, have been released. And now what happens again? What's the whole point? Increased blood glucose levels. So what happens here? Glycerol can jump into this process and it can reversibly turn into glucose. It can jump into this process here. Now, this process, which I haven't yet spoken about, is called glycolysis. Glycolysis is taking glucose to produce ATP, the opposite of what we're talking about here, right? Glucose, turning into ATP. Glucose goes to glucose 6-phosphate. Glucose 6-phosphate turns into pyruvate through a number of steps which I haven't mentioned. Pyruvate can jump into the mitochondria, turn into acetyl-CoA, and through the Krebs cycle, also known as the citric acid cycle, can produce a whole number of products. It produces NADH, it produces carbon dioxide. These products go, specifically the NADH, go to the electron transport chain to produce a whole bunch of ATP for energy. But we can hijack this system and make it go backwards to produce glucose, to increase blood glucose levels. So glycerol can jump into the glycolytic pathway, go backwards because that's what's happening, ultimately turn into glucose 6-phosphate, turn into glucose, jump out. So glycerol from triglycerides can increase blood glucose levels. All right, that's the first point. Alanine. What can alanine do? Alanine, let's write it in red, can turn into pyruvate. Now, pyruvate is irreversible. It can't go back to glucose 6-phosphate. So how do we use alanine to produce glucose if we can't go backwards? Well, pyruvate can turn into oxaloacetate. And if pyruvate turns into oxaloacetate, oxaloacetate can leave the system, ultimately turn into glucose 6-phosphate, which is turning into glucose. Brilliant. So through this process, amino acids such as alanine, not all amino acids, but alanine specifically, turn into pyruvate. Pyruvate turns into oxaloacetate, leaves the Krebs cycle, can jump back into this glycolytic pathway, but go backwards, turn into glucose 6-phosphate, and increase blood glucose levels. Fatty acids. Fatty acids can jump into the system and turn into acetyl-CoA. Now I want you to think about this. If this process of proteolysis or proteolysis and lipolysis have been stimulated, what will happen is these amino acids that have been utilized diminish the amount of oxaloacetate because they're turning into pyruvate, which is turning into oxaloacetate, and that's leaving the system. Now, oxaloacetate is a substrate that needs to bind to acetyl-CoA to produce ATP. But what's happening here is fatty acids are turning into acetyl-CoA, so acetyl-CoA levels are going up. Oxaloacetate levels are going down because they're leaving to turn into glucose. So there's a mismatch. Oxaloacetate just starts to increase. It can't bind to oxaloacetate. So what happens when all this acetyl-CoA increases too much? Well, they turn into ketones. And so acetyl-CoA, there's not much room here, will turn into ketones, and ketones can leave the system to again be utilized as an energy substrate. How? Because it jumps back into this process and it can be used to produce ATP, ketones. All right, something else, lactate. We can use lactate as a substrate in this process. And so lactate can come in and turn into pyruvate. And again, pyruvate, oxaloacetate, turn into glucose. So this process called gluconeogenesis, now there's no room to really write it up here, but let's put it down here. Gluconeogenesis, takes all of these non-carbohydrate based sources. So what were they? So it's amino acids, like alanine. It's fatty acids, it's glycerol, it's lactate, and takes them and utilizes them to produce glucose, increase in blood glucose levels. Insulin, like I said, is a strong negative regulator of gluconeogenesis. And after 10 to 12 hours, gluconeogenesis is contributing to 50% of all the glucose that's being released into our bloodstream. Because like I said, every hour, 8% of my glycogen is being utilized until there's hardly any or none left at all. So what we're talking about here is gluconeogenesis, glycogenolysis to ultimately increase blood glucose. Now the final point I need to make here is that it's not just glucagon that stimulates this. There are other hormones or chemicals that are released in the body that can contribute to increasing blood glucose levels in times of fasting. What are these hormones and chemicals? Noradrenaline, adrenaline, cortisol, growth hormone, and thyroid hormones. They are also strong agonists to promote this process of gluconeogenesis. Specifically, glucagon and adrenaline. So adrenaline is the sympathetic nervous system, fight or flight, right? So glucagon and adrenaline, they're fast acting. They do this immediately. But cortisol, growth hormone, thyroid hormones, they're more slow acting. They take time and they can have their effects over a longer period. So this is a quick run through of what happens metabolically to your body in the post-absorptive or fasting state.